Hello everyone and welcome on this lecture on circulation. So it's called Circulation 1 because you will have actually two lectures on circulation. And today we're going to discuss mostly circuits, pressure and flow. So understanding the pipes and how circulation works. And the next lecture will be on pumps, so heart physiology, to understand how pressure is built. So what is the problem we're going to discuss in this lecture? The question is, how do organisms bring nutrients and chemicals to all cells? So we'll first discuss diffusion and its limits based on size, and then we'll understand the need for circulation, and we'll briefly look at the structure of a circulatory system of plants and how they use passive flow. And finally, we'll move to animal circulation, where we'll discuss both open and closed circuits and the implication for what is called hemodynamics. So let's start with the limits of diffusion and the need for circulation. So the main question is how, within an organism, can you move different molecules of interest? Say, for instance, nutrients. So movement of molecules within a body, in an environment, just follow the law of diffusion. Okay? So the body or the molecule can cross a body, cross a membrane, and move by diffusion. So what determines diffusion? So here I give you the law of diffusion. And what you see is that the flux, so if you want the amount of molecules that can diffuse per surface per time, is related to the concentration differential and the distance, inversely related to the distance actually, and a coefficient which is called d. Okay. So what exactly are the consequences of this pure biophysics formula, if you want? Well, let's imagine a case. You have a source. This source generates molecules, and these molecules will go to a sink, okay, where the molecule is not. So you will have diffusion right after the membrane here, and the molecule will diffuse to the sink. First problem, the molecule, if you use diffusion to transport the molecule, if you want, the molecule will always follow a concentration gradient. That's one constraint. But more than that, if we look quantitatively, based on Fick's law of diffusion, this is the shape of the concentration of the molecule, right? So you start with the source with a very high concentration, and you see that with distance from the source, the concentration drops very fast. And actually, based on Fick's law of diffusion, which is on dc over dx, what you see is that the slope is essentially the differential in concentration divided by the length, the differential in x. And what you see and notice right away is that the further away you are from the source, the smaller will diffusion be. And eventually, when you're far enough from the source, there's nearly no concentration of the molecule available, and the flux is essentially null. So what that means is that diffusion is effective only over short distances. Why? Because the rate of diffusion is inversely proportional to distance. That's this divided by dx that you see here. So let's put a few numbers on this. Let's take a quantitative approach. Here I'm looking at the diffusion time. Diffusion time is proportional to x squared. That comes from there. So if you need to diffuse over 100 microns, okay, let's say a cell or two, that's okay, it will take a second. Now you need to diffuse over a body of 1 millimeter, which is 1,000 microns. 100 seconds now is needed. But if now you just need 1 centimeter, and that's small, it's 10,000 microns. With the square here that you have with x, it leads to 3 hours. So you see that actually the bigger an organism is, the further away from being able to use diffusion as a system it is, because then the time for a molecule to be transported would become essentially infinite. So size matters a lot in terms of what type of system of transport you can use. If you're considering a hydra, like here, you have what? 2 rows of cells, essentially. The scale you can see here, it's like 25 microns. Diffusion is okay. Relying on diffusion is no problem. And most molecules will be able to diffuse in all cells fine. But now imagine we are considering an elephant. What would be the time for a molecule to go from its gut to, let's say, the brain? It's relying on diffusion is just physically impossible. So that means organisms, when they become, of course, multicellular, but even more bigger and bigger, they need to develop specific systems to move things. And moving nutrients and waste, that's a circulatory system. That's what we're going to see today. But you need also, of course, adapted systems for respiration, for excretion, etc., etc. So that means the scale of the size of an organism both impose a problem, which is they can't rely on diffusion and they need to rely on something else. So what is something else? Well, something else is simply to have a circulatory system, right? So what does that mean physically? Why does having a circulatory system help prevent this problem, circumvent this problem? Well, it's because we replace a physical principle, the principle of diffusion, by another physical principle, the principle of bulk flow. So what is bulk flow? It's essentially what happens when a fluid goes in a pipe. So what I'm saying is that organisms, they use a system of pipes to now transport their molecule because this bulk flow in pipes is way, way, way faster than diffusion and is not altered by size the same way. So essentially, bulk flow for long distance is the circulatory response to the problem of diffusion. So what does that mean? That means you will have vascularization, you will have vessels, so pipes essentially, and that will allow to use bulk flow to solve this size problem. Okay? So we'll see different examples, but I want to mention right away the similarities and the differences between plants and animals. Both use a circulatory system, and circulation of a liquid solution, so you will have a liquid solution, will carry key molecules, for instance, nutrients, in different parts of the organism. So that fluid is called sap for plants, and it's blood and lymph for animals. The movement will happen in a pipe. That's common to plants and animals, even if then there are differences. And the key engine is not concentration. So you remember for diffusion, concentration is what matters. Well, in that case, it's pressure gradients what matters. And the key aspects to remember is that vascularization and bulk flow downstream of pressure gradients is way, way, way faster than diffusion over long distances. So if I simplify what I'm saying, to go fast and transport nutrients and molecules easily, you need 1, a plumbing system, and 2, a source of pressure, and that's true in both plants and animals. So now there is a fundamental difference between plants and animals, and I want you to really realize that. Plants use both negative and positive pressure circuits. So negative pressure is essentially suction. Positive pressure, you need a pump, something to put pressure and generate the circulation by bulk flow. Animals only use positive pressure, and the pump, you already know that, is a Hertz. So let's now start to go in detail about plant circulation. So we need a pipe system and we need a way to generate pressure. Let's start with the pipes. So pipes in vascular plants are of two kinds, the xylem and the phloem. The xylem will be responsible for the movement of water and minerals, while the phloem is responsible for the transport of organic materials. Okay? So water and minerals have one circuit, organic materials have another circuit. And maybe a way to memorize that is that phloem transports essentially food, so organic material. So where are they located, these transport systems? In herbaceous plants, xylem and phloem are kind of bundled together in what is called a vascular bundle, where you can see that the pipes are parallel to each other and together. In woody plants, that's a bit different. The xylem is essentially the circles of wood, and at the very periphery of the plant, you have this layer of phloem that goes there, and they're separated by the cambium, which are essentially plant stem cells. Okay? So xylem and phloem work very differently, so please do not confound them. The xylem has unidirectional movement. That means that the bulk flow always goes from the root to the leaves. Okay? That's what is depicted here. It goes from the roots to the leaves. So that's always the case. Phloem is very different. And why? It's because the way pressures are generated are very different between the xylem and the phloem. In the phloem, the bulk flow can be bidirectional. Why is that? It's because it always goes from tissues that are called source, and those source generates organic compounds, and the flow goes to sinks or tissues that are called sink, and those sinks essentially upload those organic compounds. So the flow always goes from source to sink, but if tissues that are source become sink, or tissues that are sink become source at different periods of the day, different seasons, then the flow will change. That's why we say the flow is bidirectional, because it all depends where the nutrients are uploaded in the phloem and where the nutrients are uptaken from the phloem. So let's go a little bit more in detail, and let's start with xylem. So what is the xylem structure? What's a pipe? The xylem is actually made up of cells that die. They induce programmed cell death, so it's not a living tissue. It's more of a structure built from dead cells. And so that structure includes two bits. Some are called tracheids. They're elongated bits of pipe. And you have vessel elements that are shorter, only exist in angiosperms. And vessel element and tracheids are the two cell types that compose the xylem. And both of them have slightly different influence on circulation that I want to discuss. So vessel elements, contrary to tracheids, they have something called perforation plates. And perforation plates are what you see here. They're essentially holes that allow to build a continuous pipe. So each vessel element is a cell transformed, and you can put many vessel elements one after each other. Thanks to these perforation plates, you get a pipe, essentially. Tracheids are fairly similar. However, they are a little bit different in the sense that they have a primary wall made of cellulose, but they have also a secondary wall made of lignin. And what that means is that the surface of the pipe is non-uniform and there's pits, and we'll see that it has an importance. So, in the xylem, if we consider an angiosperm, you will have essentially different kinds of pipes, some built by vessel elements, and some built with tracheids that have lignins. So why does it matter? Well, those two types of pipes, tracheids and vessel elements, they are complementary systems. The vessel elements with their perforation plates, they're essentially big pipe of water that allow fast transport, but they require constant tension to maintain the flow. What that means is that if at the end of the pipe you drop the pressure, all the water falls due to gravity. Okay? So that's good to have a fast pipe system, but that problem is limiting. Tracheids are a bit different. They have a high surface to volume ratio, always the surface to volume ratio, and thanks to those pits and that non-uniform lignin in the tracheids, there's a lot of surface of contact with water. What does that mean? That means that through capillarity, they can hold water against gravity. You know, like there's just so much surface that there's adhesion when transpiration is not occurring. So, if I summarize, two types of pipes compose the xylem. One is kind of like the highway, fast, but with difficulty in maintaining water if there's no transpiration. And the other one, the tracheids, hold water against gravity when transpiration is not occurring. So how do you get bulk flow in the xylem? You need a pressure. And in that case, the pressure is negative. So there's suction coming from somewhere. The theory that describes that is called the cohesion tension theory. So you have transpiration in the leaf is essentially the engine. It's what generates the pressure. And then water cohesion pulls water from the shoots to the roots. Okay? So what is that cohesion? Well, you know water is polar, right? And that allows for hydrogen bonds to occur between the molecule of water. That's what you have depicted here. So that means those molecules try to stay tight and there is cohesion. So if you pull on xylem sap somewhere, thanks to this cohesion, you will generate flow along the column. So the question is, how is this negative pressure or tension generated normally? Well, transpiration is what generates the tension that pulls on this column of water. So let's see how that works. We need to go in the leaf. And in the leaf, you know that there are those small pores called stomata. You see a stoma here. And you can have evaporation of water from the leaf in the environment. This evaporation is essentially what generates the whole phenomenon that generates the bulk flow in the xylem. How does that work? Well, if you have evaporation, transpiration, that means water vapor diffuses outside via the stomata. If you do that within the leaf, well, that vapor needs to be replaced. So in structures like microfibrils that are within the leaf, some water is lost by evaporation. And that means the water film here, the interface between water and air, is essentially decreasing. If you keep evaporating and decreasing, that water interface with air starts to be pulled because it interacts with the microfibrils. Again, it's capillarity, right? So simply, if you want, you increase tension at this air-water interface due to the fact that you lose water to evaporation. When you do that, by increasing tension, surface tension, that pulls on the water. And if you start pulling on the water here, because it's a continuous water circuit, and because you have water cohesion, then it pulls and generates negative pressure, suction, on the xylem, and then you have bulk flow. So transpiration leads to increase in surface tension that pulls on the water of the xylem, and that's how you have negative pressure in the xylem. So let's now discuss phloem structure and understand how phloem works. So the phloem translocates products of photosynthesis, so for instance, think carbohydrates like sucrose, from source tissues to sink tissues. The pipes themselves are fairly similar in structure from the xylem. For instance, you have sieve elements, so cells, that compose bits of the pipes, and they're connected by pores, again, called sieve plates, and that's how you build pipes. One difference is that surrounding these pipes, so these sieve elements, you have companion cells. And companion cells are the cells that transport the sugar, so meaning either they load it in the phloem, or they take it from the phloem and load it on the sink tissues. And that's what we'll see generates the bulk flow in the phloem. So let's discuss that a little bit more. How do you get positive hydrostatic pressure in the phloem? And so please remember, there's suction, so negative pressure in the xylem. In the phloem, it's positive pressure. So we transport phloem sap, which is a solution of water, but also sugar. And the sugar will be transported from a source. So imagine, for instance, mature leaves who do photosynthesis, to a sink. So for instance, imagine a flower, a fruit, a root, or things like that. However, change the season, and it's very possible that the root, for instance, loads sugar on the phloem, and the directionality would be reversed. So remember, phloem is bidirectional. So how does that work? Where does the pressure come from? Well, the movement through the phloem occurs thanks to something called translocation. Translocation is what? Is the loading of sugar by companion cells at specific sites of the phloem. So let's take an example. Let's imagine that here around the 1, you have a source cell. Say a leaf that is generating sucrose. Okay? The companion cells will load sucrose in the phloem, and you will have here a high concentration of sucrose. If you do that by osmosis, you will bring more water in the phloem at the level of the source cell. If you have more water, the pressure increases, and therefore you generate a gradient of pressure with high pressure around the source cell. That's what is written here. Water follows the sugar by osmosis, increasing hydrostatic pressure. And if you think about it, at the sink level, at the level of cells who take sugar from the phloem and within the tissue, you have the opposite. Less sugar means less water is kept, water moves out by osmosis, you have a decrease in pressure. So here what you have is that the loading of sugar at the level of the source generates a pressure gradient and therefore bulk flow. All right, so xylem is suction initiated by transpiration in the leaves, and in the phloem you have positive hydrostatic pressure due to loading of sugar and osmosis generating this local high pressure of water. So let's now move to animal circulation. Animal circulation will have only positive pressure, but we will find some open and closed circuits, and so I want to discuss that. So any circuit will have a circulatory fluid, all right, a set of vessels, and a pump. And the pump is the heart. That's what drives pressure through the system. We will have open and closed circuits, and we will have variations around the theme of a closed circuit, as we'll see today. So what is an open circuit? So that's typically a circuit you find in invertebrates. So think crustaceans, insects, mollusks, etc. So you have the pump, the heart, you have a few vessels, but they are not connected in a closed circuit. Still, the heart generates some flow, some mixing occur in the body cavity, so the hemocoel, and that fluid called hemolymph circulates like that throughout the body. It's very different from a closed circulatory system. In a closed circulatory system, the blood, so the fluid here in circulation, is closed at all times within vessels, and vessels vary in size along the circuit. So you have a continuity between the heart, the pipes, and come back to the heart. That circulatory system needs to have exchange with an interstitial fluid in which the cells of your body reside. Okay? So here we take an example of an annelid, for instance. But so the blood does not fill the body cavity. The body cavities are filled with fluid, and the blood is kind of a different compartment on its own. Very different from the open system, where the hemolymph goes in circulation, but also surrounds all tissues and fill all body cavities. So how does a closed circulatory system look like? Here you have like the basic elements. You will have three types of vessels. Arteries go from the heart to the periphery. Veins go from the peripheral tissues back to the heart. And capillaries will connect arteries and veins together in a continuous system. So that's what you see depicted here. You have arteries and arterioles, who are just smaller versions, and then they connect with the vein system through capillaries, and capillaries are the place of interface with most tissues. Where does the pressure come from? Well, you need a pressure generator, that's the heart. And the heart is essentially, in most animals, two types of chamber. You will have atria, and you will have ventricles. So the atrium, the first type of chamber, here depicted, collects the blood, and it primes the pump. But the main pump, what really puts pressure in the circuit, is the ventricle. So the ventricle is a thick-walled structure. You see, it's a strong muscle that generates most of the pressure to push blood in the circuit. Amongst organisms with a closed circulatory system, you have different designs that exist. And I want to discuss with you 4 types of designs. And those designs are interesting because they demonstrate a coevolution between the circulatory system and the respiratory system. So let's start with the first type of design that you find in fish. And this design is called single circulation. So in fishes, you have one heart that has one atrium and one ventricle. That heart pushes the flow in circulation. A stands for atrium, V stands for ventricle. All right. You will have 2 systems of capillaries, one around the respiratory system, so the gills, and one around the rest of the body, so tissues. And this heart pushes blood flow from the gills to then the body, to then the heart. So you see that the heart has to put pressure on blood so that it goes through 2 layers of capillaries before coming back. Okay? And please note where the blood is rich in oxygen, so that's red here, or poor in oxygen, that's blue here. So all structures are lined up in a single circuit, and that's a lot of effort for the single atrium and ventricle to go through these 2 layers of capillaries. So another design, slightly different, is to now have double circulation. So in amphibians, you will have 2 atria and 1 ventricle. What is the invention here? The invention is the invention of lungs and double circulation. So this only ventricle, the 1 ventricle, puts pressure on the whole circulatory system. Okay? It pushes blood both in the lungs and in body capillaries. But you see that there's not 2 successive capillaries layers. It's just the pressure of the ventricle is exerted both on capillaries of the lungs and of the body. The flow comes back through distinct atria and distinct circuit. So you have a pulmocutaneous circuit that goes back to the left atrium, and you have a systemic circuit that goes to the right atrium. And you can see, of course, the blood coming from the lungs is rich in oxygen, and the blood coming from the body is, and the peripheric tissues is poor in oxygen. Okay? So pressure is maintained along the circuit to go both towards the body and the lungs. But you see that here, the blood mixes and is partially aerated. So the fact that the blood rich in oxygen and poor in oxygen is mixed in the ventricle is kind of suboptimal. But thanks to those 2 systems, 2 circulatory systems, you don't have to go through 2 layers of capillaries. So then you have reptiles. In reptiles, the design is the same, right? You have lungs and a body. You have 2 atria, 1 ventricle. However, you separate now pulmonary and systemic circulation with a separation within the ventricle. So the ventricle is divided, if you want, in 2 halves, and 1 half pushes blood towards the lung, and the other half pushes blood towards the body. So there's no more mixing of the blood, and that really is more efficient system. And now really the blood poor on oxygen goes to the lungs, and the blood rich in oxygen goes to the body. However, you still have only 1 ventricle. The last design I want to consider is what there is in birds and mammals. And here now you have 2 ventricles, so 2 atria and 2 ventricles. And that means that you can put pressure by independently to the pulmonary and to the systemic circuits. And this double circulation also means that there will be 2 passages through the heart for each cycle, right? It allows higher blood pressure because you have 2 pressurization system, right? Right ventricle, left ventricle. And it's important as the lung has a high surface and many capillaries, so that could be a loss of pressure. So at the end, this double circulatory system with 2 atria and 2 ventricles is the most efficient in terms of loss of pressure and having efficient circulation through both the respiratory and systemic system. So in mammals, I just want to zoom. How does the circuit look like? Of course, you need to know that. So there's 2 independent capillary circuits, right? 1 around the lungs, 1 systemic around the rest of the body and the tissues. For each cycle, you can go through the whole flow. You see that there's 2 passages through the heart. For instance, if we start here, from the tissues, you go back to the heart with the blood poor in oxygen. the heart sends that to the pulmonary circuit. In the lung, the blood is loaded with oxygen, and now the left heart pushes that in the systemic compartment. So pulmonary vein and systemic arteries are rich in oxygen, and systemic veins and pulmonary arteries are low in oxygen, right? So make sure you do not mix and match that, and make sure you know, so veins and arteries, and where is the respiratory system, where is the rest of the body, and where is rich and poor oxygen levels. So the left heart in such a system controls the systemic circuit, that's what put pressures here, and the right heart controls the pulmonary circuit. So here I give you another schematic description of this kind of circuit. You see here the left heart, so in red the systemic circuit that goes to the tissues, and then the veins go back to the heart, right heart, then going to the lungs, getting oxygen and circulation here. So what I want to mention on this diagram is that 70% of the blood in circulation resides in the vein compartment. So those are called capacitance vessels. That just means that most of the capacity of the blood volume is in the veins. 16% is in arteries. Another one thing I would like you to really understand is that the number of vessels change while we move through the circuit. And with the number of vessels, inversely changes the typical diameter, of those vessels. So let's go through the circuit. For instance, if we start with the left heart, you have the aorta. You have one vessel that gets the entire circulation coming from the left heart. And the diameter is 1.1 centimeter. We keep going through the circuit, and then you will have some arteries. You will have around 10,000 arteries. But their diameter decreases to 0.5 millimeter. Keep going through the circuit, you have arterioles. And you have 10 million of those arterioles. But their diameter is only now 15 micrometers, so very small. And finally, you can go to capillaries, and you have 10 billion capillaries, but that are only 5 to 9 micrometer of diameter. So why? That's because you have an arborization. We discussed that before, and it's typically the case that you have an arborization of vessels, but because you have more and more vessels, you have also lower and lower diameter. And we'll see that this is extremely important to understand flow through a closed circulatory system. Of course, then the opposite, the symmetry happens. You see that you go from many vessels with small diameter to fewer and fewer vessels with higher and higher diameter. So why does it matter? Well, let's do a little bit of hemodynamics. What is that? It's the physics of blood flow. And I want to discuss the relationships between blood flow, resistance, pressure, and velocity. So velocity is how fast the blood goes through a pipe. Pressure is obviously positive pressure coming here from the heart. And resistance to that pressure, and how all of those determine blood flow. So the first thing to understand is that a vessel is nothing else than a pipe. Okay? There's a radius or a diameter, and there's length. And how blood flows through that pipe is just governed by simple physics. So fluid mechanics applies. Blood is a viscous fluid, and that means it's moving in the pipe by a phenomenon called laminar flow. There's no turbulence, and essentially what laminar flow is, if you consider this is the pipe, is that the fluid will flow in parallel layers without disruption, and at the center of the pipe the flow is very fast, and the closer the layer is to the pipe, the slower the movement is. So the velocity of the external layer is low, and the velocity at the center of the pipe is high. So this is called laminar flow, so flow in different layers. Okay. So what controls laminar flow is called Poiseuille's law. And I want you to understand at least a few aspects of Poiseuille's law and how they influence the physiology of circulation. So what does Poiseuille's law say? It says the flux, so how much blood goes through a pipe, depends on the differential in pressure, of course, more pressure, more flow, multiplied by 0.4, a constant, but what really matters is that it's multiplied by the radius of the pipe to the power 4 and divided by viscosity, that's also a constant in our case, and the length of the pipe. Okay. So this term, 0.4 by the radius to the power 4 and divided by viscosity and the length, is essentially a term for resistance. So another way to say that is to say that the flow rate, called Q, is equal to the delta in pressure, so pressure, divided by resistance. Okay? This is a trick for you for memorizing that, is that Q equal P divided by R. So the way to write it is P equal QR. So if I ask a little question about that, you can always say, okay, PQR, and then you know that the pressure equal the flow rate multiplied by the resistance, and you can find each other. So what that means is that that flow rate depends with pressure, so higher pressure, more flow, higher resistance, of course, less flow. And the speed, the velocity for a given flow is that flow divided by the cross-sectional area. Okay? So these three aspects are important, and you will see why. One thing I want you to notice is that the resistance is in 1 divided by R4. So what that means is that the radius of each pipe will influence resistance of the circuit, and therefore will influence the relationship between flow and pressure. All right. So what does that mean concretely? You understand PQR, you understand velocity. What does that mean? Well, let's have a look at circulation. We said here, for instance, you would have arteries, you have this arborization. Here you have many capillaries, and then you have veins. As an example, if we depict here the systemic circulation. Well, you see that if we look at the entire circuit, at different positions of the circuit, the diameter, or the radius, changes massively. Here you have many vessels with small radius, while here you have only one vessel with one big radius. That will change things a lot just based on Poiseuille's law. So microcirculation is circulation in the smallest blood vessels. So around here, the capillaries, if you want. So of course, at different levels of the circuit, the net flow, the entire flow, is unchanged, right? If there's a flow here, there's the same flow here, and there's the same flow here. But the linear velocity of the blood will change. Why? Because velocity depends on flow but surface area, and the surface area change. The cross-sectional area change. So that makes what? That makes the fact that blood flow will strongly decrease around the capillaries because the cross-sectional area will strongly increase around the capillaries. So the arborization, as a consequence, is that around the capillaries where arborization is maximal, the cross-sectional area is maximal, and the blood, for a given flow, moves very slowly. So let's give a few examples. Let's imagine here it's the aorta. Okay, you have 80 centimeter cube per second of net flow. Here, there's a capillary. You have also 80 centimeter cube per second. That's the same flow. However, here, the velocity is 20 centimeter per second, while here the velocity is 0.02 centimeter per second. Okay? So you can see that basically what it's saying is that for a given constant flow on the circuit, you use many more small pipes here, and that means the blood flows way slower. Why does it matter? It's because here that allows interaction with the tissues. Remember, the capillaries are wrapped either around the lungs or around your tissues, and you need exchange, right? So where you need exchange with the tissue at the level of capillaries, the blood is very slow. And just by the design of the circuit, where you need no more exchange, you have less vessels and therefore much more velocity. So tissue perfusion, which is the name of exchanging these nutrients with the tissue, or gas, occurs at the microcapillary sector where the blood is slow. So let's discuss and zoom a little bit more on this microcirculatory sector. So you have capillaries depicted here, and arterioles, which are like the small segment of the arterial compartment, connect to the capillaries. And what I want you to know is that arterioles control the flow of blood within the capillaries. If there's no specific signal, arterioles generally maintain the blood flow stable. How is that done? Well, if there is distension of vessels due to, let's say, blood pressure, that will trigger smooth muscle contraction, and you see that around the arterioles there are smooth muscles. And what that means is that normally, if there's no external physiological signals, the diameter or the radius of those arterioles and capillaries will remain fairly constant. And if it's constant, then blood flow can remain constant. That level of contraction of the smooth muscles that surround the vessels is called vascular tone. So higher tones means more contraction, means smaller radius. And without any physiological regulation, those muscles try to keep the radius constant. However, there can be, and there often is, regulation and physiological regulation of circulation. So how is that done? It's done by changing the vascular tone of the arterioles. So let's take an example. For instance, we consider an artery here, a capillary here, and you have this arteriole and this arteriolar vascular tone. If the tone increases, you contract the smooth muscle, you decrease the radius of the arteriole. If you decrease the radius of the arteriole, you kind of block the flow, right? So the flow in the capillary goes down. In the compartment upstream, you actually increase the resistance to blood flow. That's what it means. So through changes in vascular tone, you can change how much flow there is in the capillary, and you can change the resistance of the flow in the artery. That's why arteries and arterioles are called resistance vessels, because they determine the resistance of the network. You can see it alters blood volume distribution, right? Like you have more or less blood in the downstream or upstream compartments. And this regulates the pressure in the system, because if you increase vascular tone, you see that here you will have more pressure in the upstream compartment. So if vascular tone stays constant, you see that it maintains the flow. But if physiological regulation make it change and you contract or relax more those smooth muscles, you will control vascular flow in different capillaries. So how does it work? Well, for instance, let's imagine that a tissue requires more activity. For instance, the skeletal muscle are generating some activity. They require more blood flow. Well, the vascular tone can be affected and regulated by nerves and hormonal signal. So if there's more activity in the skeletal muscle, for instance, you could open the capillaries, decrease the vascular tone around the tissues that you want to irrigate, so for instance the skeletal muscle, and now more blood will irrigate this tissue. That's called perfusion. So you will increase tissue perfusion. The opposite is possible. If this tissue does not need a lot of blood flow, nerve and hormonal signal could increase vascular tone, decrease blood flow and perfusion, and now the blood basically, essentially, does not irrigate really those tissues. So regulation of vascular tone allows to decide where the blood flow is distributed. It's a regulation of blood distribution in the body. So let's tie things together now with this microcirculatory flow and how it's regulated. So if you don't have any physiological regulation, you have a normal arteriolar tone. So what that means is that the smooth muscle around, just make sure that the radius, the diameter, stays constant so that the flows is uninterrupted. But you can sometimes vasoconstrict, which means increase contraction of the muscles, decrease the radius or diameter of the pipe, or you can vasodilate, decrease the contraction of the muscle, increase the diameter of the radius, based on physiological regulation. So nerves and hormones can manipulate, alter vasoconstriction and vasodilation. And when there's vasodilation, you bring more blood flow to a tissue. When there's vasoconstriction, you bring less blood flow to a tissue. So then one of the questions you could ask is what regulates that? Well, the levels of oxygen, carbon dioxide, and the function, for instance, of a tissue, could regulate vasoconstriction, vasodilation. So demands of the tissue, physiological demands, could regulate vasoconstriction and vasodilation. However, you could also wonder, but why not always have maximal flow? You could always vasodilate every capillary, every arteriole in the body, and then you would always have maximum blood flow, and that's perfect. Well, that's physically impossible. And vasoconstriction and vasodilation need to be coordinated. What that means is that they need to occur in some sites in your body, but not other. For instance, if you digest, you will vasodilate in the digestive tract, bring more blood there to support the energy of digestion, but you will vasoconstrict in the muscles. If you now make some physical exercise, you will vasodilate in the muscles, but you will vasoconstrict in the digestive tract. Why? It's because of Poiseuille's law again. So let's just consider the entire circuit. We said the rate of blood flow depends on the pressure and the resistance. Okay. And the flow rate, well, you don't have choice. It's the cardiac output. It's how much the heart can deliver. So you have a given constant flow rate. And now here I put the formula with the pressure and the expression of resistance. So you see here, again, this radius to the power 4. So imagine that in the entire circuit, we decide to vasodilate. We make that radius very big. With a power 4, you would have this value be extremely high. So that means the resistance would essentially completely drop. Right? You vasodilate everywhere, the resistance drops massively. If the resistance drops for a given flow, that means physically the pressure drops. So if you were to open and vasodilate all the capillaries, the blood pressure would drop. And if the pressure drops, well, of course, then you would not be able to bring blood to specific tissues, like for instance, to the brain. You would decrease perfusion, and that's what is called a distributive shock. Right? So if you apply Poiseuille's law to the entire circuit, you realize that you cannot open and vasodilate everywhere because you would lead to a drop in pressure. So what has to happen is that vasoconstriction and vasodilation need to be regulated, and when you vasodilate and bring more blood to one sector of the circuit, you need to vasoconstrict and bring less blood to another one. All right, so next lecture will be dedicated to the pump and heart physiology. It was great discussing circulation with you guys. Have a good day and bye.